you are able to see the slides. So are you able to see? Ping it. Okay, let me see. Are you able to see the slides?
Yes, ma'am.
Okay. Okay. So let's put it on a slideshow. It's quite an interesting session because we are speaking about the pharmacovigilance. Because whenever we are speaking about the drugs, we need to ensure the safety, and the safety comes from directly pharmacovigilance, and especially when the drug is in the market. This is the only field which ensure the safety till the time drug is in the market. So that is the reason it's given so importance. And also in a clinical trial, if you see function wise, the pharmacovigilance is the biggest function, although there is a bit of a saturation in this field. So that's why I'm sharing this, because many a times the pharmacy student, they think so that okay, only pharmacovigilance is the field for them. But I just wanted to share with you, you can go beyond this field, right? You can try medical affairs, you can try even your medical writing, even clinical trial transparency, then maybe move to pharmacovigilance after some time. So the whole idea is that in case that initially you feel that it is bit struggling for you to transition into pharmacovigilance, no worries. You have many other options in your hand, even operations. Although you have done this course, so don't limit yourself. So the post-marketing surveillance. So the journey of any drug. We come from a lab, and then we go for animal testing. Then you file IND application. Then we of course go for your phase 1, then followed by phase 2, followed by phase 3, right? Then now NDA, right? New drug application. So once this is done, then you can get your product into a market. Now when it is in the market, you have to ensure the safety of the drug, right? So you have to ensure the safety of a drug. There are two ways we handle it. One is hypothesis generation. Second is we go with the hypothesis confirmation. First of all, we just go with an idea that this may be possible. One of example is the rare incident, rare side effect. We think that there might be some. Then maybe one incident in 1 lakh, this is coming. This is like maybe based on our hypothesis generation. The confirmation is when we will be having a data. So this is how we work on the post-marketing surveillance studies. And initially when we are having a patent application with us, then there is a lot of stress on this. Lot of studies are designed for this. Once patent is over, we go for BAB studies, then it fall into a general category. So till the time the pharma company is having a license with them, they also are responsible for a pharmacovigilance plan they have to submit. So this is the whole story of your post-marketing surveillance. Now, what we see in hypothesis generation is a spontaneous reporting, intensified ADR reporting. Of course, you know what ADR is. Just ping me what ADR is. Adverse drug reaction. That means there may be a possibility that some relation to a drug, it may be caused by the treatment or a drug, right? That is why the D is their drug. Don't get confused it with AE or SA. We also do the targeted reporting. We will speak about it. Cohort event monitoring, quite popular. Now, under hypothesis confirmation, because now we want to confirm it. That is case control studies. maybe globally I will show you the template of case control studies, don't worry. So case control studies, suppose globally we got 15 case. Cohort studies, randomized clinical trial, basically they planned a trial after the product is in the market. Meta-analysis studies. Now we will speak about each method in detail. Let me see if there is some question or concern. Okay, all going well. So pharmacovigilance method, we are having a passive surveillance. Passive means we are not making any active effort. The system is there, and if it came to notice, then someone is reporting based on their awareness level. So this relies on data collection from a disease observation on ad hoc basis. Most usual form of surveillance undertaken in the most countries. Globally, the most popular surveillance is a passive surveillance. Now, under this also, there are two type of further divisions, you can say the further type of reporting. One is spontaneous reports. Second is case series. The spontaneous reports are like, suppose you are a pharmacist. I know many of our students, they belong to pharmacy, right? You are a clinical pharmacist in some hospital and you saw this is something is not mentioned in the package insert, right? And you thought to report it. Correct? So that is spontaneous report. Case series. You at a level 2, you developed a series of cases, like one person came. I will also show you the narrative like in a case series. So hold on. One person of 60-year-old came in. You prepared whole series in that type of a case, right? So the spontaneous reporting system, let's speak about first spontaneous reporting system. The system to monitor the safety of all medicine. So big, small, high risk, low risk, your vaccines, everything falls under. So by default, this system applies to every, what you say, drug or medicine out there. This is a voluntarily submission of case report by healthcare professionals or pharmaceutical manufacturers or patient to the National Pharmacovigilance Program. It is just like that India will be collecting its own, Japan will be, I'm just taking few examples. These are countries from Asia, similarly from EU, say France, they are collecting, UK is collecting at own, US is collecting at own. All these spontaneous ADRs, they will get into a global system. Suppose India reported 1, Japan reported 3, France reported 5, UK nil or like that. So it will give a fair idea that how much this drug is safe globally. This is how it goes. There is a report and data entry will done. What is the report? Like suppose an example, some person taken a vaccine. All of a sudden that person started feeling lot of itching and nurse was standing there. I'm just telling you in the form of story. Nurse was standing there. Nurse saw that this is not a usual. This is different. Then what she will do? She'll put it in a system. What she will put? Important information like indication, name of drug, date, maybe some information about the patient. And this is important, right? She will enter and then you, the pharmacovigilance associate, this case will come to you. This is the initial information came. Now here you will give it MEDRA terminology. You will do coding assessment. If some further information, suppose here she just added the name of a drug, maybe manufacturer, other details she has not added. You will complete it. Then it will be lead to a signal detection. Signal detection is not only, I'll come to that. It will lead to a signal detection, like related to fever, related to itching. Here are 100 events reported globally. Here are 150 events reported globally. Right. This will came into our system. This is giving us some signal. Signal is nothing. It is just a new information related to that drug. That's all. That's the whole story. So maybe India, we reported only one incident of a fever. But when we saw the system globally, pharmacovigilance system, we saw that it is more than 100. Then what signal this is giving us? This is signal this is giving us that this drug is causing fever as a ADR. So this will become your database. This is for one thing. Then similarly for other things, other areas, it will build a data. So now coming, this is important. If some interview you face and someone asks you, what can be reported in a spontaneous reporting or what cannot be reported? The idea is anything big, anything small, it all that can be reported as part of a spontaneous reporting. So when you are confused, example, you are confused, shall I report or shall I not report? What to do? Report. And this is the training also in once you are in a proper setup. Every year you will be doing it. Serious ADRs, mandatory, non-mandatory, all you will report. Severe ADRs, new drugs, unknown, unlabeled reaction. That means it is not there in investigator brochure or a drug leaflet. It was not there, but it will report it. ADRs in a vulnerable group like children, pregnant women, elderly, they are all reported. Okay, what is serious ADRs? That is, life-threatening. It is an extension of an SE. Fatal, life-threatening, maybe causing a permanent disability, your prolonged hospitalization, or any medical significant event. So that will get reported under spontaneous reporting. So this is good example where you can see and analyze that more a kind of a decision question. If you wanted to reach somewhere, serious reaction, report, yes. If it is no. Say example, this came to you, is a serious reaction. If yes, report. Now your analysis saying no, it is not a serious. Involve medicine on additional monitoring. If this answer to second question, does it involve medicine which require additional monitoring? Report. If this question is also no, unlisted reaction means it was not there in a investigator brochure or patient drug leaflet. Unlisted, yes, report. Even if it is not, so it was not unlisted as well, it was there either in IV or a drug leaflet. Next question is, does it involve child or any vulnerable population? Report yes. If this is also not sure, you are not sure that if it's involving being a pediatric population or any other vulnerable group, what is, you are not sure, basically is confused state, not to know what to do, right? Report. So idea of whole this decision question is to at the end analyze, do you report or do you not report? The crux of the whole story is you report. It is the later team of a medical affair. If you get to work as a medical affairs, they can analyze, they can at a later stage, at an advanced stage, they can say that because there will be a decision question related or not related. If they're choosing not related, it will come in the category of AE, adverse event, not in ADR. But that is not a judgment call when we see any serious reaction. So we have to just, this is very simple. This is very interesting and but very simple. You are more kind of a dealing in a yes or no, and you are reaching at a final stage. Any question on this diagram? You see it, just paste your question here if you are unable to mute. Okay, I believe we are good. Silence. So now, what is spontaneous reporting good at? First of all, it covers whole population. We need not to set a special system. It covers all race, ethnicity globally. Number two, all medicine. This is covering all medicine. It's not selective. Continuous monitoring throughout the life cycle of the drug. It's not like a planned part, like when NDA is approved, maybe till 5 years of pharma companies observing it. It is not like that. Till the time the drug is on the pharmacy stores, this system will going on round the clock. Detect signals, new, rare, and serious areas. So this is interesting. Although spontaneous reporting is a voluntary reporting, but the most important and the rare side effect they came into known to know us is by this system is the biggest example that given a birth to a pharmacovigilance is thalidomide tragedy. How this came to notice is just because a doctor he prepared the case report and he published it, right? Again, and falling under the scope of spontaneous reporting. Most commonly used method, no doubt, easiest method to establish. Don't need lot of infrastructure, least labor intensive. Nurse, doctor, pharmacist, they are doing their own regular day-to-day job, and suddenly they are spotting something different, unique, they are ready to report. No expense, relatively very inexpensive, because now if you see in the advanced set of system globally, most of the countries they are having a national pharmacovigilance programs. Like India also has all the tier 1 leading hospital, they are having a pharmacovigilance function in them, including our these private hospitals. What are the problems with the spontaneous reporting? Underreporting, because it's more on a voluntary basis, that if someone feel the need, they will report. If they see that there is no need, they may skip it. Capture it only suspected areas that we are suspecting. Sometimes it get biased about the seriousness and the severity. New medicine, people pay more attention when there is a new drug in the market, instead the one which is existing since years or decades. And advertisement of the product, that can also affect publicity of specific areas. Denominator is known. Suppose you have seen one case or maybe 10 cases, suppose an example in India. Now, what denominator I will give it to you? Like, have I analyzed 1,000? Have I analyzed 1 lakh population? What it is, so that the proportion or the percentage, the stat value is not coming out of it. Difficult to detect, delayed reporting. Now, another bit advanced version of a spontaneous reporting, because we are talking about hypothesis generation. We are trying to see this may exist. So this is basically enhanced ADR reporting of a specific medicine, early reporting phase. Suppose year 2023, I have launched some vaccine. Maybe for next 5 years, till the time this is in the market, we are educating different hospitals, healthcare professionals, like, look, this is a new vaccine. If you see something different about it, report it. This is an extension of spontaneous report program, right? Basically, we are making people more aware about it, that be little more cautious about this new product, new drug, new vaccine, new treatment when you administrate it. So that is intensified. So basically, medicinal product is subject to additional monitoring. Now the targeted reporting. This is again in our hypothesis generation. This is one little more advanced version. This is intensified ADR reporting within defined cohort. Suppose we have defined a specific population. It may be a race, it may be an ethnicity, it may be a gender. Like, say example, in defined cohort, take it, the topmost layer is a male and female, right? This is how we see the effect of drug. So we just say we wanted to target only female. They want to see that maybe there are suspicion of ADRs is more, right? So that's how you see. Another example, you can say the specific population, patient living with HIV infection. Again, we defined a cohort. This is, we have defined it. Cohort is defined that only folks who are having HIV infection. That is your defined population. Now you will also see the regional PV centers, specific medicine. Suppose example, now you see tenofovir. This is one medicine. This is causing lot of renal toxicity. This is another way to define or target something, that okay, this medicine you're looking, just check for the renal toxicity. So now you can also define, look at this big blue picture. Excuse me. That specific areas. Do you want to target specific areas? All areas, specific medicine you want to target, specific clinic also, hospital also, specific population also. Specific ADR is also, for you are giving just take an example of this rotavirus vaccine, because it helped to prevent the diarrhea in infants. But if the child, because of the vaccine, child is experiencing a lot of diarrhea, that means it is her ADR of our interest, because we are giving this vaccine to prevent it, but if same time it is causing similar effect, then need to check. Then in that case, they check the viral shedding in a stool. I mean, this is little advanced, but we are targeting, we are focusing on it, right? Target spontaneous reporting, that this incident may be 1 in 200, incident may be 1 in 1 lakh, we don't know. Serious ADR are captured, treatment duration, long-term duration, if we want to know that what is the effect of long-term duration. What are the problems with the target spontaneous? What can be, you can say that best things happen with the target spontaneous reports. Can utilize the existing reporting infrastructure. We need not to develop new, as I told you, or the big leading hospitals. When we undergone this new drug rules 2019, India paid a lot of attention to develop a pharmacovigilance infrastructure. Mostly globally, people are using WHO systems. So that is a bit of difference. But because of our population load, India has a population load, these systems are not that workable in our population, because number is always high. So that's the reason we have to work on a different plan. But again, if we are talking about targeted spontaneous reporting, you can utilize already existing infrastructure. This can target specific medicine of interest, possible to implement the monitoring plan that targets specific issue of concern, maybe specific area or medicine, patient group. Like best is, this slide is the best one, the way I have given you example. But then capture useful information, less background noise. Denominator is known, because when you are targeting some population, say we have taken patient with HIV infection, you can collect the data, you know the disease burden, right? What are the problems? Underreporting. Any system with a spontaneous word is underreporting, is always is the problem. Capture only suspected area, known toxicity, because we are focusing on something, like I said, rotavirus, diarrhea. That this drug, we are focusing on renal toxicity, where our focus is narrow. May limit reporting only specific areas, relies on diagnostic capability of a reporter. If pharmacist or a doctor or nurse, they miss it, they think, no, according to me, just giving you an example, definition of a diarrhea reporting is more than three. They are saying that more than 3 stools a day is the definition of a diarrhea. But some nurse may say that, oh, 3 is okay. Maybe someone can do, otherwise, 2 stools, that person can report it is a diarrhea. So that capability, again, the capability plays a role. Now we are moving to another pharmacovigilance tool, which is a cohort event monitoring. It's very interesting way to collect the information. This gather more information on a safety profile of a new chemical entity. So now see, because we are working on the hypothesis generation. Hypothesis generation will be more for the new drug, right? Suppose this year, 2024, like Novo Nordics last year, they launched their Ozempic for the diabetes and all that, right? So they just launched in 2024, and now they will be focusing on here for this medicine till at least 5 years or 4 years. What are the side effects? After coming into the market phase, of course, we look all these side effects when the drug was there out in the clinical phase, but now we are focusing once in the market. What they will do, because now see this Ozempic medicine drug, which is in news also, it has given a kick to their share also, right? You see, they will be planning observational studies, observational cohort study design. That's when we give this NDA application to any regulatory body, we have to give them a plan for the post-marketing surveillance. Now, patient will be enrolled in these cohorts, actively followed up during treatment to record the all adverse events. Because now you see lakhs and lakhs of people, they are taking this drug. In clinical trials, maybe 1,000, 2,000, 3,000. Take an example of 10,000 max, that is the highest limit, right? But now there is no limit. Additionally, the people living with other health conditions, like, okay, diabetes also there. Now that person may be having a BP issue, maybe taking some other medication. So they have to plan and observe cohort. They will make 2 groups, like, okay, group 1 taking this medicine, drug A, group 2 taking some other drug. This will become control. They will compare the effect of these two. These will be more on observational studies. Like this will be the cohort, they will make population differentiation. Now, what are the known reactions? They characterize the known reactions. They try to detect the signals of unrecognized reactions and they identify interaction with other medicine, very important. And this is with your other TCM is your other traditional medicines, your Ayurvedic, all that. Say, take an easiest example. Say we have introduced new vaccine for infants, because infants, they undergo a lot of vaccinations when they are small. So this new vaccine, what they will see in the cohort event monitoring or the pharmacovigilance program on the vaccine in the market, one is how it is interacting with other childhood vaccines. Say it is a polio vaccine, which is very commonly used, right? Or maybe typhoid vaccine, how this new vaccine is interacting with it. Because we are introducing this new vaccine, because of this new vaccine, if it is decreasing the effect of a polio vaccine or a typhoid vaccine or measles vaccine, then what is the impact? Because these old vaccines, they are already part of very established system. So that is your drug-drug interaction, right? It's an easy way to put it like this. Detect inefficiency of medicine. We know the safety is our priority, right? That's why we have a pharmacovigilance. However, the efficacy is how well this drug is working. If this medicine is not bringing effect, if I'm taking a drug to take care of my fever, but it is not helping, that is inefficiency of that drug. That should also be reported. Or it may be otherwise also. There are many drugs that we introduced into a system for something else, and they are also helping us with. Take very easy example, this retinol people are using as a part of a skincare. More for an acne, then later they realize, let's use it for anti-aging, because that came to know because of our pharmacovigilance reporting system. Assessment on the pregnant and lactating mothers, any other risk factor. This is more a comparative slide, bit an overlap, but important is here to focus. Spontaneous reporting, the crux is denominator unknown. We take a suspected ADRs and it cover all medicine, not only all medicine, all population it covers. Minimal reporting, WHO has a program for international drug monitoring, right? So, but as I have shared with you that India has to, ICMR develop their own systems because of our population. Intensified ADR reporting, but they are all systems that we have developed. Just wanted to tell you, they are all aligned to WHO programs or international programs, right? They are not different. Intensified reporting, again, denominator, unknown, suspected areas, specific medicine. We can focus on specific medicine. And it is helping us with early post-marketing drug phases to be launched here, and they can help us. Targeted reporting, denominator, again, unknown, suspected areas, specific area, cohort-specific medicines. Cohort event monitoring, denominator unknown. All events, it cover all events. Again, this cohort event monitoring and intensified ADR reporting is especially for the post-marketing surveillance. When the new drug is in the market, for a couple of years, we run these programs. I can take a pause. You can just take a look by yourself, on this, because we have done with it. This kind of a comparison. All right. So I guess we can move for next is case series. For hypothesis, we are moving now to hypothesis confirmation, right? Let me see if you are having any questions. Okay. So case series, because we are now trying a part of hypothesis confirmation. This is collection of case reports that share some common characteristics, such as being exposed to same drug and in which same outcome is observed. So they are collected globally. In case series involve reports of 2 or more people with common exposure to drug and common outcome. Both people taken same vaccine. Say example, they have taken vaccine for diarrhea, and they are getting seizures. Infant, I'm speaking about the infants. The drug is also same and the outcome is also same. Another example you can take is the polio vaccine they have taken, same vaccine, but the outcome is maybe headache. The same outcome. That is your case series. Now 2 cases reported from India, 5 cases from other parts. That's all. It becomes series. Series of case reports provide evidence and association of a drug and adverse event. Suppose one child experiencing a seizure, standalone case. Another child experience, that is adding into a fact. Used generally for generating an hypothesis and then verifying the association between a drug and exposure outcome. What are the advantages? Easy to write. I can write a case, generate hypothesis. Observations are useful for other researchers. Disadvantages: biasness, lack of comparison group. So I have, based on standalone cases, I reported the case, but I don't know how it stands with the control. There is no control to compare. Now the case report, this goes on a level 2. Case reports are the description of history of a single patient who has exposed to medicine and experienced particular unexpected effect, whether the effect is beneficial or harmful. So this is the, I will show you one example of, you can also say the narrative, patient safety narrative, right? Single history of one patient, that is your report. Now the history of multiple patients will make a series. Case report has a privileged place because it may be the first signal for an adverse event. This is the best example. You are thalidomide tragedy. So now the sanitary sites. People think this is quite difficult. No, it's not difficult. So they are choosing a specific hospitals, clinics, or any healthcare site where they think the prevalence of this particular disease will be highest. Say example, they like our aims in Delhi. It is like go-to for most of pharma company. Why? The kind of patient population is coming there is no other site can match. Like Rajiv Gandhi Cancer Hospital in Delhi. If there is anything related to oncology, cancer, that site will get selected, right? So this is active surveillance. We are intentionally doing it. We are intentionally planning it. Sanitary sites we are selecting can be achieved by reviewing medical records or interviewing the patient and physician in a sample of that particular site to complete an accurate data of ADRs. The selected site, say example in a Rajiv Gandhi Cancer Hospital, we just wanted to focus on a lung cancer. All the patients, those are coming in for having a diagnosis of a lung cancer, they will get interviewed, they will get their data will be collected. The selected site can provide information such as data for a specific patient subgroup. Now in lung cancer, you wanted to select, male with history of, you can say, are very commonly smoking. Another category they can select is male with a history of not so active, mostly sitting job, not active. Like these, they can make a subgroups. Weakness of this type of system, again, biasness, small number of patients. How many patients you can review? Increased cost, because you are selecting particular site. If I'm selecting particular site, I need to select investigator also. We need to give them some facilities as well. They're investing time in to collect the data, and to collect the data they need some tools, maybe case report forms. So that is a cost associated there to it, but it is a good system. So this system is efficient for drug use for institutional settings such as hospital, nursing homes, dedicated infrastructure and reporting. Now we are moving almost to our end of our session. The registries, like there may be a registry on a cardiology, there may be a registry on a respiratory disease, because the respiratory one is the There may be a registry on diabetes. So what is this registry means? Suppose, just take an example, we have a registry on hypertension, which is, there are a lot many, right? You are sitting here in some city, you saw one case of hypertension that came into notice. You add into this registry, you will add. Similarly, people globally will keep on adding. There will be day, this number go when grow to 10,000, 1 lakh, 1 million, all that, right? And then you will be having a data in this register online to compare. Like the way, can you see the importance of a clinicaltrials.gov, or it has grown? If you go, the number of trial that get registered on that website is high. So it is becoming an analysis sheet for others. So this is how the purpose, they have to set the registry. The registry is a list of patients presenting with the same characteristics. So example, I have given you hypertension. Now there are another registry can be on a breast cancer, another registry can be some childhood vaccine, another registry can be on a diabetes. There can be endless registries, and if you google it, you go to your, any, you will be able to find so many registries. And if you're planning for your phd or a master's thesis, the registries are going to help you a lot for your meta-analysis and other things. A disease registry such as for a blood, anything. I will, these are just some examples, because basically you have to see the drug exposure and how it is associated with your clinical condition. A disease registry can also be used as a base for a case control study comparing with the drug exposure cases that are identified. Uh, drug registers, similarly, drug register can be also be made. So there is a registry for a rheumatoid arthritis patient exposed to biological therapies to determine if the drug has a special impact on this particular group of a patient. Some registries address drug exposure in a specific population. Example, pregnant women, very popular group, very popular group. Patient can be followed over a time and included in a cohort to collect, because for a pregnant woman, see, anyways, their journey of 9 months will be followed, right? So that's how this easy to collect the data. Okay, and there are always standardized questionnaires. Now, on the comparative observational studies, these are all the planned studies. You can say that these are epidemiological methods to collect the data. Planned comparative studies, suppose we have a drug A. This is our new drug. It will be compared with a control. That's it. This will be group A. This is one group, this is second group. So we are comparing a new drug with a control. Now this control can be the patient who are not taking drug at all. This may be group who are taking standard of care, right? So that can be. So this is epidemiological method and the key component in evaluation of adverse event. So there are a number of observation study design that those help us to validate the signals. If you are, just try this activity. A new drug is introduced to a market. A has new mechanism of action compared to other drug of a market. How would you monitor the safety of drug A and why? Okay, I think I will try to show you this one thing. This is what we can say the case series or type of thing they prepare in a, say, drug, they will just insert the name of a drug. What is the suspected ADR? It may be a hepatotoxicity, liver toxicity, database source, VisiBase, Vigilance. These are very popular, and the date of analysis. So what? See, this is just I try to make it like a table: case ID, then age and sex, then the country, indication, dose, time of setting, seriousness. So the first case is 45-year-old male coming from UK, fungal infection. This is like a kind of hypothetical dose. Additional information, alcohol use. Another, and the second case, 60-year-old female, nail infection, fatal, quite a fatal disease. The first one was hospitalized. Another additional factor, she was also taking paracetamol and she's also diabetic. Another case from US, case number 32, year 32, female, US, ring bomb, non-serious, but her additional condition, none. So this is how they analyze the cases globally, right? Now, for a narrative for a single event, this is how we generally, this is very small example. We generally try to write a narrative for a one incident, like a 60-year-old female from India treated with a drug X for nail infection, developed acute liver failure after 5 weeks of treatment. She had diabetes and was taking paracetamol concurrently. The event was fatal. No re-challenge attempted. This is basically known as narrative. Narrative is nothing but is the story form of your AESA. So that's all from my side. I hope this was interesting for you guys and help to gain you some knowledge. Are you able to understand at least not 100%, maybe 50, 20%, 30%? Was it clear to you? Silence अच्छा नहीं है ना? Silence is quite deadly. Okay, thank you, thank you, thank you. We will be having, I guess, I'm not sure if you are part of internship. Are you a part of internship program also? We will be having this week, I guess, informed consent form. Yes, okay. Please do attend. We will try to do multiple activities. I intentionally requested to organize on Sunday morning at 11. Samriddhi has agreed to it. The reason is Sunday evening, mostly students are busy and energy is also low. I am speaking about myself. My energy sometime in Sunday evenings are low. So for internship, I just wanted to run few exercises. We can also make a group that how to do a consent. Are you ready for it? We can even spend more time, more than 2 hours. Right, but come into that session, please. I request you, because it will help you throughout your career. So on Sunday morning by 11 we will start. Then later you are free, you can do anything, whatever you want, right? Okay, so okay, you can take more time, Vaishnavi, no problem, take more time, but do it. And for doing also, it's not like you have to do it up perfectly. Idea is you should know the tools, you should know the techniques, you should know the nitty gritty. Idea is to take out the fear from you. Okay, so no problem, you take more time. I will tell Samriddhi they need little extra time. Then we will meet on Sunday, huh? All right, take care, guys. Thanks, Rachna. Thanks, Ruchi. Thank you, guys. Bye-bye. Good night.